Hello viewers, welcome to the course of Introductory Plant Pathology. Today we will be discussing about the Mycoplasma or Phytoplasma. Mycoplasma or Phytoplasma were earlier known as MLOs, that is Mycoplasma like organisms, and these are wall-less plant bacteria under the family Molecule, and these plants are actually transmitted through leaf hoppers, and they cause different types of symptoms in the crop plants. They are wall-less plant pathogenic bacteria, and the diseased plants reside in the seed elements of the infected plants, because in the vascular bundles of the plants there are xylem and phloem. These are the vascular conduction tissues, and phloem has two types of components, that is companion cells and sieve tubes. So, these mycoplasma reside in the sieve tubes only, they are confined to the sieve tubes, and phloem feeding insects, mainly leaf hoppers, and transmit phytoplasmas from plant to plant. The phytoplasma are pleomorphic in nature, that is they are having the various variations in the shape and sizes of the cell, and with small cocoid bodies, ring forms, pear shaped, and rarely filamentous also. They are found in different shapes. In view of their infective spread, symptoms and their transmission by insects, MLOs were previously considered as the viruses, and many disease symptoms of the MLOs are mostly akin to the symptoms caused by the viruses. So, attempts were made in the Mycoplasma infected plants to see that whether they are from the viruses or not, and so many virological techniques were applied to see the nature of the microorganism, and it was failed totally. Then until 1967, the Mycoplasma was regarded as the viral diseases. In the year 1967, the Japanese scientist Doi et al. And Ishii et al. They worked on the Mycoplasma, and they nullified the first time the roles of viruses in the Mycoplasma diseases, and they showed the involvement of these in the causing yellow disease in the plants. They observed MLOs in phloem elements of plants infected with mulberry dwarf, potato witch's broom, and ester yellows. Usually mycoplasmas are non-motile, unlikely of the bacteria and fungi, and filterable through bacterial filters. They are very small, and their diameter ranges from 0.3 to 0.9 μm. Affected plants exhibit symptoms suggesting a profound disturbance in the normal balance of the growth regulators with yellow symptoms. Detection and characterization of phytoplasmas infecting different plant species are now possible with the molecular methods based on the study of 16S rDNA polymorphisms. Mycoplasma bodies disappear on treatment with the tetracycline antibiotics, and this phenomenon is known as remission. Remission means temporary disappearance of the symptoms. Actually, what happens that when we spray the oxytetracycline on the body of the plant infected with the Mycoplasma, we find that after some time the symptoms of the Mycoplasma are disappeared. We think that it has been managed or it has been controlled, but not. This is only the remission of the symptom, and after some time the symptoms again reappear. And more than 300 distinct plant diseases have been attributed to phytoplasmas, affecting hundreds of plant genera. Now, we come to the Mycoplasma disease. Several hundred important food, vegetable, fruit crops, ornamental crops and timber and shade trees are affected with these organisms. Ester yellow's phytoplasma causes major economic losses of vegetable crops like lettuce, carrot and celery. Ornamental plants like gladiolus, hydrangea, China aster and purple cornflower, and peanut and sweet potato witch's broom, sesamum and soybean phyllodes cause considerable losses of the crops in the Asian region. These are the most important diseases caused by Mycoplasma. They include spike disease of sandal, witch's broom of opuntia, corn stunt, coconut lethal yellowing, grapevine yellows, sesamum phyllodes, grassy shoot of sugarcane and brinjal little leaf disease. Then symptoms and transmission. As we have noted earlier, that profound disturbance in the normal balance of growth regulators leading to the virescence or phyllodes. Phyllodes is nothing but the green leaf like structures are formed instead of flowers. And this I would like to tell you that normally auxins are the bioregulants or growth regulator, which is responsible for the growth of the apical tissues and flowers and buds. But in the case of Mycoplasma, what happens that auxins are protected by the phenols, and these phenols act as auxin protectors by the way of increase in the oxidative enzymes like IA oxidase, polyphenol oxidase and peroxidase. These types of malformations or proliferations of the tissues are commonly seen in case of Mycoplasma affected plants. Proliferation of axillary bud shoots changing in witch's broom symptoms. This is the proliferation, means the malformation of the tissues, and sterility of the flowers, abnormal internode elongation, generalized stunting with yellowing or reddening of the leaves, decline and death of plant, and phytoplasmas are transmitted by insects belonging to the family Cicadelidae, Cixidae, Psyllidae, Delphasidae and Derbidae. These are the insect vector responsible for transmitting Mycoplasma from plant to plant. The Nephotetics, Cincticeps and Ampoesca fabi are the main insect responsible for this transmission. This is the picture of sesamum phyllodes. You see the second and fourth one showing the healthy plant which is having flower and fruits, but the phyllodes affected plants are showing the malformation and typical type of growth on the apical side, having no flowers and no fruits. This is the sesamum phyllodes in the severe case. You see the reddening of the leaves is very common and a bushy structure is formed under the severity of the infection. Likewise, the little leaf disease of eggplant, you see the leaves are reduced considerably in comparison to the normal leaves, and no flowers and fruits are being formed, and under the severity of the infection you see the whole plant has turned into a bush like structure. Then this is the flower virescence of the periwinkle. Actually, virescence is a simple phenomenon which shows that in place of the color, the green color is dominating in these affected plants, and here you see the periwinkle plant is having this purple color which is being turned into the green color, green patches are seen. It is a virescence of the periwinkle due to the Mycoplasma. and in this is the lime tree showing the witch's broom infections, and this is a very advanced stage of infection showing the drying of the bush, and ultimately this will die off. Then this is the Stolber phytoplasma affecting tomato, showing the upper leaf and axillary buds affected with the Mycoplasma infection. This is the ester yellow phyllodes on the coneflower, and you see the coneflower are so beautiful, having the purple and pinkish purple color, but the affected plant is due to virescence, showing the green flowers turned into green leafy structures. And this is the sugarcane grassy shoot. This is economically very important disease of Mycoplasma, because it affects the sugarcane plants, and instead of developing the cane in the field, we see the proliferation of the tissues and bush like structure of the plant. The internodes are becoming short, and the plant is showing the white and yellow leaves without producing any cane. Yes, now I would like to discuss more about the Sesamum phyllodes. This is a very important disease, as I told you earlier, and very often we find the sesamum, the oilseed crop of our country, is affected mostly by Mycoplasma diseases. And we have seen that earlier times, when we had no much facilities about the biotechnological tools to study these diseases, we used to read the functions of these diseases under microscope in the laboratory, and we have devised certain stains known as Dian's stains, I may say, which can be used for detecting under the compound microscope. When we use this dye, certain Mycoplasma get red stain, and we can see under the microscope the presence of these bodies under the laboratory conditions, if we do not have the much improved and advanced instruments with us. You see, this is a very important, as the whole plant is converted into a bush like structure, and we find that around 30 to 40% incidence of this disease is there in the farmer's field, especially in the arid western area of Rajasthan and other states of India. Now, we come to the little leaf of brinjal. This is also a very important disease, and as I have mentioned here, that it has been first reported from India by Thomas and Krishnaswamy in the year 1939, but they were not aware that this disease is due to the Mycoplasma, but they thought that this is due to the viruses. And this typical symptom of this disease shows that how much virus and Mycoplasma are related to each other. They are also not found in the meristem, and this is also absent in the meristem. And in for developing the resistant plants, we can use this technique through plant tissue culture, that we can excise the meristem of the affected plants, and if we culture this meristem on the plant tissue culture media to raise the plants and organize these plants, we can have a plenty of chances to have the healthy Mycoplasma free plants in our laboratories. This Phytoplasma disease, as we have seen, that ornamental plants are much more affected in these disease affecting areas, and you see the virescence and the affection on the lime, which is the most important economic tree of the horticultural plants, and tomato is the most important vegetable crop. They are very sensitive to these type of diseases. Then aster yellow, which is an ornamental plant, as I have shown you that this type of flowers, when affected, they are totally useless for the growers, as they do not find any kind of flowers in the affected plants. As we have seen, the beautiful flowers of the cone flowers are converted into the leaf like structures with the green virescence, and they show that the value, the ornamental value of the plant is greatly reduced by the incidence of the Mycoplasma disease on such ornamental plants. Likewise, I may again reiterate that the sugarcane grassy shoot disease is very important for our country, as this is increasing day by day, and it is covering the large areas in our farmers fields growing sugarcane for production of sugar, and by way of doing this, we can see that if we are not able to have the managing strategy for this type of the pathogens, it will be difficult for us to cover this area in for sugar production, typically caused by the Mycoplasma. Then we come to the classification. The most comprehensive and widely accepted classification scheme is based on restriction fragment length polymorphism, that is RFLP analysis of polymerase chain reaction, that is PCR. This approach provided a reliable tool for broad differentiation among phytoplasmas. The system has classified phytoplasmas into 19 groups and more than 40 subgroups. Following the application of molecular technologies, the enigmatic status of Mycoplasma like organisms amongst the prokaryotes was finally resolved. This led to the application of the name Phytoplasma and eventually to the designation of a new taxon for these organisms named Candidatus phytoplasma. This is the classification of Mycoplasma: kingdom Prokaryote, division Tenericutes, class Mollicutes, order Mycoplasmatales, family there is one family Mycoplasmataceae, and genus Mycoplasma. Then we come to the reproduction of Mycoplasma, that how they reproduce. MLOs can reproduce by binary fission, by formation of spores, elementary bodies, and filamentous growth and budding, as we have seen in case of bacteria. The chromosome replication starts at a fixed site followed by bidirectional progression. The process of Mycoplasma cell reproduction has not yet been well classified. Translocation in plants. Phytoplasmas are transferred by insects to plant sieve elements in the phloem tissues. They spread systemically through the plants using sieve tube system without affecting the meristems. This is the main difference between the Mycoplasma and virus. Whenever we used to free the plants from the virus, we take the meristem. So, the meristem is the free from the virus. Phytoplasmas are pleomorphic and sufficiently small to pass freely through the sieve pores. So, they may be swept along the assimilate flow from the leaves to sugar consuming plant organs. Consequently, phytoplasmas have been found in sink tissues such as immature leaves and roots, whereas source leaves remain uninfected. These are the pictures showing the different types of Mycoplasma molecules. You see here it is in the sieve tube and Mycoplasma bodies are there. It is in the sieve tubes and you see the different circular and molecule shape in the sieve tube, showing variation in the size and shape of phytoplasma in all the three pictures. Then interaction with the host. At present, exploration of the interaction of phytopathogenic molecules with their plant and insect hosts is a very fascinating and very active field of research nowadays. Several lines of investigations are underway to determine the interaction of phytopathogenic molecules with both plants and insects. Now, we come to the work of Professor Karl Marmorosch. He is a legendary person in the field of virology, entomology and mycoplasma. He was born in January 16, 1915 in Vienna, Austria. He is a renowned American virologist, entomologist and plant pathologist. It is a very rare combination in a single person. He obtained first evidence that certain plant pathogens multiply in plants as well as in specific invertebrate animal vectors. In 1956, he first cultured insect cells for use in the study of viruses. He has been an active, innovating and inspirational pioneer and contributor to the field of phytoplasmas. He used electron microscope to detect and characterize viruses and phytoplasmas in cells of diseased plants and insect vectors. He visited Indian universities also, and over 60 years he has written 800 papers and 100 books. It is such a commendable job he has been done by Professor Karl Marmorosch. Today, we discussed about the mycoplasma like organisms or the mycoplasma or phytoplasma. This is a new group of plant pathogens. Earlier, we used to call them viruses, but since 1967 from the researches of Doi et al. And Ishii et al., we came to know that this is a new group of plant pathogens which are causing variety of diseases which are not viruses in nature. They are cell-less bacteria, they are not motile and their transmission is through the insect vector leafhopper. We have seen the economic importance, the symptoms of the disease. We have seen the how much losses they cause to the economically important plants like sugarcane, like brinjal, like sesamum and all that. And ornamental plants are also very much affected by these pathogens. And we have also studied a lot about its nature and its transmission. Lastly, we have seen the exemplary work done by Professor Karl Marmorosch, who has given a commendable job by working on both these viruses, mycoplasma and insects also. Thank you.